So I want to start off here, chapter 9, which is titled Adaptation from Genes to Traits. If you were using the second edition to our evolution textbook, if you got a hard copy or whatever, then this is treated as chapter 10 in that second edition. So it's chapter 9 in our third edition. But this chapter is dealing with the evolution of complex phenotypes. Phenotypes that very traditionally would be argued to. Okay. So we're headed into chapter 9, which is titled From Genes to Traits. If you're using a hard copy or some other source of the second edition of this textbook, then this is published as chapter 10 in the second edition. They moved it into chapter 9 in the third edition. So just in case, so you know. But many aspects of organismal phenotypes are, of course, complex and historically have not been easy to explain in terms of their origin and evolution. And this is what Chapter 9 From Genes to Traits attempts to do, is to show how complex traits can evolve. One classic example of a complex trait would be the eye, which has numerous components and parts to it, and a functional eye needs all those components to be functioning properly for an eye to work. And that is, one of the challenges that evolutionary biologists have faced is how do we explain the evolution of such a complex structure? The evolution of the eye will be addressed later in this chapter. The introduction to the chapter talks about venom delivery systems in snakes specifically. And of course, we've got this beautiful image of a rattlesnake agape and ready to inject its venom, at least it looks that way. And the introductory story is about Brian Fry, a biologist at the University of Melbourne in Australia, since he studies the evolution of snake venom. And any venom delivery system can be considered a complex adaptation. And we'll address what a complex adaptation is on the next slide. But when you look at the whole venom delivery system, we've got cells that are expressing venom genes to produce the venom. We've got muscles that are operating to push on the glands to push the venom out. We've got tubes that the venom is delivered through, and those tubes, in the case of a rattlesnake or any snake, runs through the fangs. So there's numerous components to, in this case, the venom delivery system. How did venom evolve? How did venom evolve from non-venomous lizards? And in general, how do adaptations emerge and change over time? So we'll look at a few examples in this chapter where genetics, as usual, has revealed the origins of complex adaptations and has shown us that explanations that once seemed daunting and potentially even impossible now are being revealed through molecular research. I've included a link here on the slide. It's a 14-minute video on Brian Fry's research, and you might enjoy watching that just because it's pretty cool. So let's continue on and see what complex adaptations are formally considered and how they evolve. So let's address then what a complex adaptation is. So we've got a formal definition here on the top of the slide. complex adaptations include suites of co-expressed traits that together experience selection for a common function. So some examples, well, let's get some examples down below, but we can consider phenotypes to be complex when A, or blue asterisk, they are influenced by many environmental and genetic factors. They're not simple traits, they're complex in this regard. And B, or the second blue asterisk, multiple components have to be expressed together at the same time for the trait to function and work. So examples include a cobra's venom delivery service, or rattlesnake, or other snake. An eye, which I mentioned on the first slide. Or the network of proteins, for example, that produce blood clots. When you look at all of these things, there are several environmental and genetic factors at work to express these. And they all have to be expressed together at the same time for venom delivery system to work, an eye to work, blood clotting to work. So those two components that were mentioned just above with the blue asterisks, those are working for all three of these examples: venom delivery, eye, blood clotting. So genes involved in complex adaptations, we can basically divide them into two different categories. We can take the genes and say, okay, they're either genes that are encoding specifically for proteins that physically constitute the adaptation. These proteins are important for making this physical trait, this phenotype. And then we also have genes that are regulatory. And so the other category of genes involves those regulatory elements of those genes, plus the proteins and the RNA molecules that interact with those elements. So just to summarize there, we've got genes that are physically making the proteins that make up that adaptation, plus the genes involved with making proteins and RNA molecules that are interacting together to help express those traits. So we end up with a very organized hierarchy where just a few elements trigger a cascade of gene expression, one after another, and sometimes branching out like a spider web as a network, sometimes involving hundreds of genes. So again, this is getting back at the complexity of these traits. So what's the adaptive significance of this? Well, these kinds of cascades enable cells to produce intricately choreographed responses very quickly and very reliably to a relatively simple external signal. So think of the venom delivery system. So these cascades of gene expression are, intricately choreographed, expressed very quickly, very reliably to, for example, inject venom into prey or as a defense into a perceived predator. So that's what complex adaptations are. And thinking about cascades of gene expression being involved to express these adaptations is an important thing to think about as we move on here. Speaking of cascades of genes, let's take a look at this bacterial spore example. So you may recall that some types of bacteria can produce spores or endospores that can go into dormancy when environmental conditions get kind of harsh. So there you go, what I just said there, environmental conditions get kind of harsh. So there's some kind of environmental trigger that causes this cascade of gene expression to take place that results in the formation of spores. And this figure, which you can find in your textbook, by no means do I expect you to know the intricate details of this figure. But you can see by looking at it for a short while or a long while, whatever you choose, that it's pretty intricate. And indeed, it's like it is a flowchart, and it is showing the cascade of genes that are expressed when that environmental factor triggers this to happen. So first of all, we've got, as I mentioned, this whole cascade is triggered by signals, environmental signals, that are received on the cell surface. So that's where it all begins. And these signals either promote or repress the expression of genes. Both promoters and repressors can happen and do happen in these cascades. And this, of course, leads to spore formation. And the end product of this whole cascade is a dormant bacterial spore. And this bacterial spore is put together or synthesized by, number one, proteins that physically make up the spore, and number two, regulatory networks that control spore formation. So think back or go back and look at the slide that we just came from, because both of these components, number one and number two, were mentioned on the previous slide. There's two categories of genes that go into these complex adaptations. Ones that are involved with the expression of the proteins that physically make up the complex adaptation, and then the regulatory genes and networks that control how this happens. So both of those components are at work in this example with spore formation in bacteria. So still on the subject of the cascades of genes that are responsible for these complex adaptations, let's look at an animal development example. So you've heard about, you know about Hox genes. Hox genes are a class of homeotic genes, genes that control the development of body parts and the placement of body parts. Both plants and animals have homeotic genes, and the Hox genes are unique to animals. No other life forms on the planet have Hox genes specifically. So Hox genes help determine what the different segments in a fly embryo, or any insect for that matter, but a fly embryo, will become in an adult. Some of these Hox genes shut down other developmental genes. We could take a look at figure 9.3. I apologize, I still have figure 10.3 listed on this slide. And it shows you, again, it looks pretty complex, but we can boil it down here or distill it down to what's going on here for our example that you should be able to describe or express. So we have this developmental gene called distalis or distalis. And of course, this gene, like any gene, is found in every single cell of the fly's body. And we have another gene called homothorax, abbreviated HTH, homothorax. So those are the two key genes that we're looking at, developmental genes that we're looking at for this example, to show how these two genes in various combinations control the development of certain body parts going on certain places of the fly's body. So for example, if the gene distalis and the gene homothorax, HTH, if they are both expressed in certain cells, then antennae will form. If distalis if distalis is expressed, but homothorax is not expressed, then legs or mouth parts will develop. If distalis is not expressed at all, then we won't see any appendages develop. And this is what we see on the abdomen region of insects. The abdomen has no appendages coming off of it, whether it's a fly, a grasshopper, a cockroach, whatever. And so these combinations of gene expression and the cascades of expressions that occur in different regions of the embryo, different cells of the embryo, are what determine the development of these major body parts. Again, it wasn't too long ago that biologists were totally at a wall of understanding how such complex parts of an animal like a fly or an insect could develop. Now we understand. Again, genetics holds the keys. So we can conclude from this example that if we understand these hierarchies, if we can pinpoint down how these hierarchies work, it will allow us, it does allow us to see how gene networks offer opportunities for evolutionary change, including shifts in body parts or novel functions, like repurposing ancient genes into new functions. Because indeed, those are the patterns that we see as we delve into the evolution of complex adaptations and using the genetic tools that we now have at our fingertips. We understand that understanding these hierarchies allows us to see how these gene networks can be modified through mutation to give rise to new types of animals with new types of adaptations. So here we're taking a look, moving on in our chapter, at how innovations, evolutionary innovations, are generated. And there's several ways by which old proteins, pre-existing proteins, and RNA molecules can take on brand new functions, forming a brand new adaptation. And that's what I'm summarizing here. That's what I'm listing here with these various blue asterisks, are these several ways that new functions can be taken on, giving rise to new adaptations. So you might remember back in chapter 5, when we were reviewing some genetics, bacteria, archaeans, some single-cell eukaryotes may acquire and recruit new genes via horizontal gene transfer. So that's one way. It can pick up some genes from somewhere else. If you're a prokaryote, you can do that through conjugation or transformation or transduction. If you're a ciliate, like paramecium, they pass on little pieces of or micronuclei from one cell to another. So there's horizontal gene transfer. So that's one way you can get some new genes and then recruit them for new functions. You can also have mutations in pre-existing genes, mutations specifically in regulatory regions. And by mutating regulatory regions, that controls gene expression and can alter pathways, can turn them on or turn them off. So the first asterisk was simply talking about how we can get new genes from outside sources. The second asterisk here, again, is just pointing out that, hey, a mutation can happen in a pre-existing gene, a regulatory region, turning on or turning off pathways. Another way is through what we call promiscuous proteins. Promiscuous proteins are proteins that actually have more than one function. So, for example, some enzymes, while we learn in our introductory biology courses that each enzyme is very specific and has just one function, in fact, in some cases, enzymes can work with different substrates, although not very well. They can be promiscuous, and they might be able to trigger chemical reactions as catalysts that on substrates that aren't the primary target. They're being promiscuous. So selected pressures could cause a protein to change its shape, through mutation, favoring one chemical reaction over the other chemical reactions. Since these proteins are promiscuous, selection could favor the promiscuity to go in one direction or another direction. And then the fourth example here, the fourth asterisk, is gene duplications. So this is going to result in what we call paralogs in terms of homology. And see the figure down below from your textbook. And so by having a gene duplication, not only do you have the original copy that can continue to be used as it always has been, but the new copy, the duplicated copy, is now freed up to perform a totally new function. The door is open to do something totally new. A great example that your chapter talks about, read the details about this, is duplications in olfactory genes in mammals. So having many, many genes that are producing protein receptors, olfactory receptors, that are able to receive different chemical signals. It's how we're able to, or mammals are able to smell, if you will, so many different kinds of chemicals in the environment, as they have all these duplicated olfactory genes that have been freed up, if you will, to be receptive to different signals. And many of those genes in certain species of mammals that don't have a great sense of smell have become pseudogenes, so they've been turned off. They're like fossils. Other types of mammals have an incredible number of olfactory genes through gene duplication that gives their sense of smell a mind-boggling magnitude. So if you compare humans, which kind of have a mediocre sense of smell, to dolphins, which have a horrible sense of smell, and elephants, which are really the kings of olfactory senses. They have thousands of olfactory genes that are all operating. Gene duplication has enabled these different lineages of mammals to have different olfactory senses. And then under that gene duplication category, gene recruitment uses a pre-existing gene in a whole new network for a whole new function. So once you get a duplicated gene, again, one of those copies is free to use in a brand new network for producing a new adaptation. So the figure on the bottom of your slide here is showing you how you could start off with a gene, protein coding DNA, that has nearby regulatory regions. And, well, that gene could be duplicated. So if you follow the figure there, now you've got 2 copies of this gene. And with 2 copies of that gene, then each of those copies can be freed up then to be performing some new functions. So generating innovations. Several ways by which old proteins and RNA molecules can take on new functions, new adaptations, and this slide is summarizing for you the myriad ways, the diverse ways that this can happen. So here we've got a great example of a new innovation being observed, being documented in real time metabolic evolution in microbes. So remember that Richard Lenski experiment, 25-year experiment, still going on today with the 12 evolving lines of E. coli? Well, we're looking at that experiment again because during that experiment, they documented the evolution of a whole new metabolic pathway that allowed E. coli to use an energy source that they were not able to use previously. So that's a pretty big deal. So normally, E. coli is only able to feed on citrate in the absence of oxygen. So E. coli feeds on glucose in the presence of oxygen, and then when oxygen's depleted, then they could feed on citrate. And at generation, well, let's say first, to show how this happens. So the first blue asterisk there, in the absence of oxygen, the CYP genes are expressed. And when they're expressed, a membrane protein pumps out succinate from the cell while pumping in citrate. So if you look at figure B, part B, or the top of the figure there, it's showing you how that happens. So the CYP promoter is activated and the CYP gene is expressed. And when it's expressed, then that transport protein does its job of pumping in citrate, but only in the absence of oxygen. At generation 33,000, they started to notice in the laboratory that one of the lines of bacteria became really cloudy, even when glucose had been depleted. And it was like, something interesting's going on here. They're trying to troubleshoot to see, was there some kind of contamination or whatever? But no, it was the E. coli that was doing this. The E. coli now were doing something different that they weren't doing before. And the solution was super cloudy because there was such a high density of E. coli. They're able to metabolize and reproduce. So it turned out that one of those lines evolved the ability to metabolize citrate and extract energy from it in the presence of oxygen. So a whole new metabolic innovation. And what was discovered was that there was a 2,933 base pair sequence that was duplicated. So check out the figure. Basically, that's part B down there on the bottom of the figure. And that duplication contained a copy of the CYP gene. It contained part of what's called the rank gene, RNK, and the promoter sequence that controls that rank gene, the expression of the rank gene. So there was a duplicated copy. And with that duplicated copy, the rank promoter was now controlling the duplicated CYP gene, and the promoter was activating the new CYP gene in the presence of oxygen. Remember, before, that was only being activated in the absence of oxygen. So now with this duplicated gene, it now was being activated in the presence of oxygen. So it's a whole new adaptation that was now allowing those bacteria that had that duplicated gene, that new adaptation, to be able to metabolize citrate in the presence of oxygen, even when glucose was absent. So it gave those bacteria an edge, a selective edge against the wild type bacteria that weren't able to metabolize in the absence of oxygen citrate. And so it opened the door for the promotion and the evolution of this new line of bacteria due to this gene duplication. The origin of a new adaptation in real time. Okay, so we just came out of an example showing how we're able to document and observe a brand new innovation, evolutionary innovation, a brand new adaptation in real time by studying microbes. Well, of course, we can't observe the emergence of a new adaptation like that in animals, like we can in bacteria, because we've got this generation time problem. At generation 33,000, we had that shift in the E. coli due to a gene duplication, and of course, that could be documented. And now they're up to over 100,000 generations, but we would not be able to do an experiment like this with something like an animal, like a snake or whatever, a mammal that has such long generation times. But we can get to the bottom of how complex adaptations can evolve in something like a venomous snake by comparing anatomy and comparing genes among related species. And by doing these comparisons can reveal evolutionary patterns and processes. So originally, we've got these beta defensins that are produced in the skin and other parts of the body for fighting bacteria. And we produce these beta defensins as humans, mammals. Mice have them, humans, baboons, wild boars, etc. So basically, producing these beta defensins against bacteria goes back quite a ways in our evolutionary history. But when we look at venomous snakes, it turns out that these beta defensin genes have been duplicated, and those duplicated copies then are freed up to be selected for brand new functions. And in this case, what we've come to learn is they're freed up and used to make venom. So about half of the active genes in venom glands in snakes, whether you're talking about a cobra, a rattlesnake, whatever, are general sort of housekeeping genes. And the other half of the genes that are expressed are actual, producing venom molecules. So if we take a phylogenetic perspective, we look at the upper right figure, top right, you have a phylogeny that's showing mammals and snakes. And it shows that the precursor to these muscle-destroying venoms that we call crotamines, the precursor to the crotamine venom was the beta defensin proteins. They are homologous, except that these crotamine proteins, the venoms, are produced in other parts of the body to fight, or the beta defensins are produced in other parts of the body to fight infections. Whereas in snakes, these have been co-opted and expressed in the venom glands to produce crotamine and other types of venoms. So gene duplication, once again, is what has allowed new defensin proteins to attack other pathogens in the pancreas. So that was the first step in snakes. If you take a look at that phylogeny on the top of the page, it shows a synapomorphy, 3 purple synapomorphies, and the one in the middle of the phylogeny for snakes says beta defensins for fighting bacteria are expressed in the snake's pancreas and other organs, but not in the venom glands. Okay, so that was something that happened within the snake lineage. Now, gene duplication, coupled with other mutations in regulatory genes, caused some of these defensins to be expressed elsewhere. Because remember, the genes found in all the cells of the body, but now mutations have happened such that these defensins are being expressed, for example, in the mouth, in the venom. And so again, we're dealing with figure 9.9, not 10.9. And so if you look on the left-hand side of the slide here where that figure is shown, we're saying, okay, we're starting off with, in the snake condition, a single copy of the defensin gene being expressed in the pancreas. And we get a duplication of the defensin gene. Okay, so that duplication is found in all the cells of the body, but it's being expressed in the pancreas. But then there's a change in gene regulation. There's a mutation affecting gene regulation. And that duplicated copy that snakes have is now being expressed in the mouth rather than in the pancreas. And as this defensin protein is now being expressed in the mouth, it is now beginning to act as a venom to capture prey. And so this is gene recruitment, venom gland recruitment. And since then, additional duplications have happened and losses as venoms have diversified in different lineages of snakes. Different lineages of snakes have different cocktails of venoms that do different things. And so that's the final sort of synapomorphy that you see on the upper right-hand phylogeny on the slide. It says beta defensins evolve into crotamine venom that is expressed only in the venom glands of snakes. So through this comparative approach of looking at anatomy and how genes are expressed in different parts of the body, to sort of summarize this, defensin proteins are already made in other amniotes, being expressed, for example, in the skin as a defense against bacteria. In snakes, the beta defensins started to be expressed in the snake's pancreas. Then there was gene duplication, and that gene duplication originally being expressed in the pancreas began to be, through a change in regulation, expressed in the mouth, the venom gland. And then that continued over evolutionary time with more gene duplications, and those genes being used to make different kinds of venoms, different kinds of proteins, producing the kinds of cocktails of venoms that are now so well known in the snake lineages. So here we can take an even closer look at the history of venom in snakes to get a better picture of how the venom and venom delivery systems evolved in snakes. So evolving gene networks in complex organisms. So there are lots of novel venoms, unique venoms. There's at least 24 that have been documented, and they have all originated from gene duplications and changes in gene regulation. And through these changes, that's led to gene recruitment. Hey, we're going to use you now for a new function, a new job. We're recruiting you. And then mutations and selection fine-tuned these adaptations, giving rise to these very complex cocktails and different lineages of venoms. So take a look at figure 9.10. Again, this is kind of driving me crazy because it says 10.10. And by the way, let me let you know, I'm narrating this with this computer system down in the Maricopa colleges, which is how I have access to PowerPoint. And that was through a cyber attack. And so I'm unable to modify this PowerPoint file at the moment, and I can only use it as is to tell you this stuff. So that's why it's like you might be wondering, well, why didn't you change this? Why doesn't it say 9.10? I'm kind of at a quandary right now, but keeping moving with this stuff. So anyway, little side story there about why I'm have figure 10.10 instead of 9.10. So if you look at the figure on the upper right-hand side of the slide, it's showing you a bunch of different snake toxins, listing them, and the effects they have, whether they're neurotoxins or decaying tissues or causing changes in blood pressure or causing muscle decay or whatever. There are numerous different effects that these snake toxins that have arisen through gene duplication, different effects they have on prey or whatever has been bitten. And you can, so some venoms have evolved before the evolution of even complex venom delivery systems came about. So for example, garter snakes do not have any fangs, but they still produce venom. And this is a relatively new discovery. It was when I learned about snake biology many years ago, it was, well, garter snakes are harmless. It turns out they actually produce a venom. And what we have now, through this comparative approach of looking at multiple more distantly related species and examining the genes in these more distantly related species, we can see that some of these venoms evolved in the common ancestor of snakes and lizards. If you look at the figure in the bottom left of the slide, it says figure 10.11, but it's 9.11. Some of these venoms evolved millions of years before snakes originated. And so, for example, some of the iguanas and monitor lizards, glass lizards, Gila monsters. We've got venoms in all these guys, and you can see that there's a bunch of venoms by the cladogram that evolved in the common ancestor of all of them. It's just taken off at an accelerated rate in snakes. And so it's through this comparative anatomy and genetic approach that we have come to discover that venom, in fact, is something that is not just found in certain snakes that we've traditionally considered venomous, but venom is also found in other snakes that have been considered non-venomous, and venom originated even farther back ago in the common ancestor of snakes and many lineages of lizards. So coming back to the topic of animal development, for many, many, many years, biologists, and again, through my early training as a biologist, I lived through this, through what I was taught in my undergraduate and even many of my graduate level classes, that while we have these different kinds of animals, like insects and vertebrates that have digestive systems and nervous systems, for example, the positioning or the placement of these body plans is very different. There are profound differences in how our bodies are put together. So while we all have nervous systems and digestive systems, how they develop and where they are in our bodies is very different. And so it was thought for a long time that perhaps we evolved these systems independently. The genetics for these systems came about independently. You know, it was a real quandary, a real conundrum, if you will. How did this happen? And what we know now, again, through some of the revelations through developmental genetics, is that we are all built with the same basic genetic toolkit. And that is an expression that developmental biologists use quite frequently, is genetic toolkit. So we're using the same genetic toolkit that our common ancestor had. We now know this. And we're using the same genetic toolkit to basically build different kinds of bodies. So for example, so that's like a statement that's being said on the slide. You know, take that and let's see an example. let's work through an example of how, while we have very profound differences in body plans, we're building these different body plans with the same basic genetic toolkits. So for example, both mice and flies have 4 homologous sets of Hox genes that are expressed from head to tail in the body. Now, in vertebrates, which of course mice are vertebrates, the entire Hox cluster was duplicated twice. The figure to the left shows this. And so we have a lot more Hox genes to work with in our toolkit for making our complex vertebrate bodies. But both mice and flies have similar microRNAs that regulate our Hox genes. So not only do we have these 4 homologous sets, but we have similar microRNAs that are regulating them. And just to show how this homology works, and how profound it is that even though we end up with such different bodies, we got these genes from a common ancestor. The Hox gene, this is in red on the bottom of your slide, the Hox gene in a fly can be silenced, turned off, and take the corresponding Hox gene from a mouse and insert it into the fly, and the fly will develop normally. So examples like this are testaments to our homology and how we have inherited these developmental genes from our common ancestors, and we use them in genetic toolkits to basically build our different kinds of bodies. So let's look a little deeper at our genetic toolkit that we use for development, whether you're a vertebrate, a mouse, or an insect, a fruit fly. Our body parts are arranged from our front end, anterior, to our rear end, posterior, so from front to back. And our body parts are also arranged from dorsal to ventral, top to bottom. And these body parts are arranged from anterior to posterior or dorsal to ventral by homologous Hox genes, determining the placement of these body parts. So the genes that we're focused on here in this example on this slide, second red bullet point, we have the 2 genes BMP4, just call it BUMP4, and cordon, and those are found in mice. Now there are homologs that are found in flies, and they go by different names. They go by different names because they were discovered by a different research team, but in fact they are homologous. This is something that kind of bugs me. Sorry for the pun. It'd be awfully nice if they were called the same thing since they're homologs. But anyway, in the fruit fly, DPP is the homolog to BUMP4 in the mouse, and SOG is the homolog to cordon in the mouse. Okay, so that's establishing the homologs. And these genes, these homologous genes, control the dorsal to ventral placement of body parts. But in a mouse compared to a fruit fly, in a vertebrate compared to an insect, they're expressed in reverse. And because they're expressed in reverse, this explains our different body plans. The question that confounded biologists for so long, look, hey, in a fruit fly, the nervous system is placed on the bottom of the animal and the digestive system, on the top. But in a mouse, it's reversed. And so, as was expressed a little bit earlier, for a long time, biologists thought, well, does this mean that these systems evolved independently? No, they didn't. We have the same genes, they're homologous, we got them from a common ancestor, but they're being expressed differently. And so because of this difference in expression, we have, for example, if you look at the bottom right figure, in the fruit fly, DPP is expressed dorsally, and so that gives the digestive system, whereas its homologue, BUMP4, in a mouse is expressed ventrally. So that body part, that body system, is put in a completely different position. And the same reversal occurs with the nervous system. So now we have an explanation for our different body plans. Genetics has once again come to the rescue, developmental genetics, to help us understand the evolution of these complex body plans in such different animals, such as a mouse and a fruit fly. So what we can see then is that all bilaterians, octopuses, sea stars, oysters, earthworms, humans, all bilaterian animals, have inherited the same networks of body patterning genes from a common ancestor over 570 million years ago. But changes in the regulation of these genes has produced different body plans. So the last thing we're going to look at here under the genetic toolkit for development, finally, for now, we're going to address this question on this slide. Can we infer the position/arrangement of the nerve cord in the common ancestor of bilaterians? So again, vertebrates, our nerve cord is positioned dorsally, and in insects, it's positioned ventrally. So now are we in a position where we can assess where the nerve cord may have been positioned in the common ancestor of all of us, bilaterians? So here we're using this comparative approach. So now we're going to bring into the picture here cnidarians. Cnidarians are the outgroup to bilateria, cnidarians being like jellies and sea anemones and corals. They don't have a nerve cord. They have a nerve net. So their nervous system is spread out like a net. We have this relatively new lineage of worms that we didn't know about very long ago that live in the deep sea, in the mud and the sediments in the bottom. And these are called xenocoelomorphs. Xenocoelomorphs. And what we've learned about them is A, they are basal in our animal lineage. Check them out in that cladogram on the slide, that phylogeny. They are basically the outgroup to vertebrates and other invertebrates. And what we see in the xenocoelomorphs is a variety of different patterns of the nervous system. Some of them show a nerve net. Some of them show some nerve cord positioning in different parts of the body. So there's quite a bit of variation. So what we can assess from that then is that the common ancestor of bilaterians probably started off with a nerve net kind of arrangement, because if cnidarians have a nerve net and some of the xenocoelomorphs have a nerve net, it stands to reason parsimoniously that a nerve net was the ancestral condition. And then if we look at the phylogeny, that includes vertebrates and arthropods and annelids and those other animals that you see in the cladogram, we see that, okay, vertebrates have a dorsal position of the nerve cord. Arthropods have a ventral position, and annelids also have a ventral position. But when we look at their closest relatives, there is no dorsal or ventral patterning in them of a nerve cord. So look at that cladogram. In the hemichordates, which is an outgroup to vertebrates, there is no dorsal or ventral patterning. They have both. Okay, so that implies that vertebrates evolved their dorsal patterning independently. Then we look down at the arthropods on that cladogram, And we see that their outgroup, their sister taxon, are the nematodes. They also do not have exclusively a dorsal or a ventral patterning in the nematodes. So that implies that the arthropods evolved their ventral position of their nerve cord independently. And then finally in the annelids, it's the same deal. The outgroup to the annelids, well, we would have mollusks, and we'd have here shown in this diagram platyhelminthes, which again, annelids have a ventral patterning of the nervous system, so they evolved that independently. So our conclusion then, through this comparative approach, is that nerve cords evolved 3 separate times. That's the most parsimonious explanation. So we're going to end this as a part 1 narration. And in part 2, to finish out this chapter, we're going to look at the evolution of limbs. We're going to look at the evolution of eyes. We're going to look at convergent evolution and a couple other topics.